we have to study the next cycle, that is hatch and slag cycle, that is C4 cycle, or it is also known as hatch and slag cycle. And this is this type of cycle is going to occur in maize plant, in sorghum plant, in sugarcane plant, in euphorbia, so these are the plants where this type of cycle occur. Actually, we know that behavior of rubisco changes when temperature is high and concentration of oxygen is also high. We all know that at this condition the rubisco start behaving as oxygenase instead of carboxylase. So to overcome this problem, plant is adapted for themselves so that the rubisco cannot act as oxygenase. And so what the plant adapted, that is adaptation of C4 plant. So what they have adapted, so first thing they have adapted that they have two type of chloroplast. They have two type of chloroplast. I have already tell you that is they show dimorphic chloroplast. The two type of chloroplasts that are present are granule and agranule. So in granule, the grana is present, whereas in granule, grana is absent, only stroma is present. And this type of chloroplast is found in mesophyll tissue, whereas this type of chloroplast means agranule chloroplast is found in bundle sheet. So this is the first adaptation that the plants show, that is they have dimorphic type of chloroplast. Now second thing they have adapted, they show the most important adaptation, that is Krenz anatomy in leaf. They show Krenz anatomy in leaf, that is cell of bundle sheet, shell of bundle sheet are going to arrange in a wreath-like manner. Wreath-like manner means in a circular arrangement they are arranged. Now if we'll see the diagram or section of leaf for showing the Krenz anatomy. Suppose this is the upper epidermis. Over here the cuticle will present. This will be the lower epidermis along with the stomata. This will be the lower epidermis, and we all know that between these two epidermis there are two type of parenchymatic cell is present, that is known as, one is spongy parenchyma and other is pellucid parenchyma, and this is the vascular bundle. Okay, so now the two type of cells are present, one is pellucid parenchyma and here the spongy parenchyma will present. Now around the bundle sheet there was a parenchymatous cell was present, which cover the bundle sheet and they are present in a ring form manner. So this is called as bundle sheet, which is present on the membrane of vascular bundle, and that covers the vascular bundle, and this circular structure is known as bundle sheet. So this is the type of anatomy that is present in the C4 plant. Now we have to study the C4 cycle, because this agranular chloroplast is going to occur in bundle sheet here, and this granular chloroplast is going to occur here. Now what happened? How this C4 plant overcome this problem? So when temperature is high, suppose the direct sun heat is coming and it is to the upper epidermis, then the heat will pass into this, that is the mesophyll cells, and the limited amount of heat, that is only appropriate amount of heat, will reach to the bundle sheet. So this condition is withheld. Now the oxygen. So oxygen is produced in bundle sheet. Oxygen is produced in mesophyll cells because C3 cycle is going to occur there. As now in this type of arrangement, C3 cycle will occur inside the bundle sheet. So the oxygen is not going to interfere here, because the non-cyclic reaction will going to occur in mesophyll cell and the C3 cycle is going to occur in bundle sheet. So the entrance of oxygen is not, the oxygen cannot enter in that cycle. So these two problem is overcome by this type of adaptation. Now we are going to see the C4 cycle. Now for C4 cycle, as we will know that C4 cycle occur in two places. So I am making one, you all know that the bundle sheet is surrounded by many mesophyll cells, but we are making one single cell, and this is the bundle sheet, suppose. Okay, this is mesophyll and this is bundle sheets. Now what happened? Now see the cycle actually. What happened? Carbon dioxide diffuses from outside into the mesophyll cells. Okay. Here carbon dioxide get mixed with water that is already present in a presence of an enzyme. I am making a asterisk here, and the enzyme that is helping in that is carbonic anhydrase. Carbonic anhydrase. So carbonic anhydrase will help in a process, and ultimately this is formed, that is HCO3 is formed. Now what happened actually, I am rewriting the reaction here. H2O plus CO2 in a presence of this enzyme will give rise H2CO3, that is carbonic acid, and this carbonic acid will going to split up into two parts and they will release H plus HCO3 minus ion. So this will be the whole reaction that is occurring here. Ultimately this is formed. Now to this, that is the HCO3, there were a component is present that is called PEP, that is phosphoenol pyruvate. So PEP will going to join it, and PEP is phosphoenol pyruvate. That will going to join it, that is it will going to join the join the HCO3, and for joining it, it also require an enzyme that is called phosphoenol pyruvate carboxylase. That is one more enzyme is required here, and that is called PEPC. That is phosphoenol pyruvate carboxylase. Now this will do the carboxylation, and ultimately after the carboxylation in mesosulfite only a 4 carbon compound is formed, that is oxaloacetic acid, and this is 4 carbon compound. This 4 carbon compound ultimately turns into another 4 carbon compound, that is called malic acid. Or maybe one more thing will happen, this oxaloacetic acid, I am writing it here, it may convert into aspartic acid. Depending upon the condition, it may convert into malic acid or aspartic acid. Both are 4 carbon compound. Now if malic acid is formed, this malic acid will diffuses into the bundle sheet. Okay, this malic acid diffuse into the bundle sheet, and now this 4 carbon compound releases 1 carbon dioxide, and this carbon dioxide with the help of here RuBP is present and Rubisco is present, so it will enter into the C3 cycle and ultimately form PGA and then PGAL, and ultimately this is the C3 cycle will go on here. Now here the C3 cycle will go on with the help of this 1 carbon atom. Now the left out is the 3 carbon, and this 3 carbon compound is pyruvic acid. It is 3 carbon compound. Now this pyruvic acid is entered into, again it will go into the mesophyll cell, and this pyruvic acid has to convert into the phosphoenol pyruvate. So here it will have pyruvic acid, and for here now 2 ATP molecule will convert it into 2 ADP molecule, and ultimately that lead to the formation of PEP with the help of enzyme. Now this reaction will go on, and here what happened, Rubisco is present in bundle sheet, so it does not come in contact with oxygen, and also in a process it does not come in contact with higher temperature. So ultimately it will continue the C3 cycle without undergoing photorespiration. So the loss that is occurred due to the process of photorespiration is overcome by this C4 cycle actually. Now, but this cycle needs more energy. As you will know, C3 cycle is going on, and we have calculated there that there 18 ATP are required along with 12 NADPH2, and here as this will go on, that is again the cyclic condition will go on for 1 by 1. So ultimately it also needed, as 2 ATP is required in one time, so this type of 6 cycle will go on. So 6 into 2, 12 ATP are required to convert this pyruvic acid into PEP. So 12 extra ATP are required. That means here we need 30. Here we need 30 ATP. Instead of 18 ATP, we are needing 30 ATP because 12 extra ATP are required to convert this pyruvic acid to phosphoenol pyruvate. But by expending this much of energy, the plant is overcoming the drawback of photorespiration. So this is your C4 cycle. Now we have to study the limitation of photosynthesis. That are what are the limiting